Well, a very good morning to all of you. This is Daryl Brookink, Executive Editor of No-Till Farmer. I want to welcome you to today's webinar. And if you're like any of us here in the Milwaukee office, you were digging out from some snow last night or this morning, and hopefully you're getting some good moisture to take you into the spring. I do want to thank you for joining us. And today we're going to focus on how to really be as efficient as possible with your fertility program. Part of that through allowing your soil's biological process to perform at its maximum effectiveness as well. And I want to let you know that today's presenters are John Mayernak, who is an agronomist and editor with FHR, and also Jeff Littrell, who is both a no-tiller and strip-tiller, and he's got about 2,000 acres of his own that he farms. He is also the owner of FHR out of Stewartville, Minnesota. Fertility is obviously a huge topic of focus for growers. It's a big line item in your operations. And we have been noticing a trend among our readers that they are looking to reduce the levels of applied fertilizer. Clearly, higher fertilizer prices and even more pressure from the general public on farmers to keep things like nitrates and phosphorus out of watersheds have been big. And so farmers are zeroing in on this issue. Before we turn over the controls to John Mayernak, a couple of housekeeping items for you. On your screen, you're going to see a kind of a control box. There is a small orange rectangular box with a white arrow inside it. You can click on that box. It will maximize or minimize that control box. And also inside of that control box, you'll see a chat location whereby you can type in any questions you might have. And so with any time permitting at the end of the webinar, we're going to look to answer as many questions as we can. Also, in the unlikely event we do get disconnected from this webinar or somehow would happen to crash or fail, just go back to the email you received and re-enter the webinar just as you did when you first joined us today. If something like that would happen, you'll be prepared and we'll all be prepared to get back in and get the webinar started again. So with that, I do want to thank you again for attending. I want to thank both John and Jeff for leading this webinar today. John Mayernak is going to do most presenting with Jeff, providing some additional commentary. And with that, I'm going to turn over the controls to John. Thank you, John.
Thank you, Daryl. You're Daryl, right?
That's correct.
All right, I want to thank everybody for being on the webinar this morning. This interest is good, and I hope that you can learn something and we can kind of tell you what we're up to. As you can see on your screen, the Earth's a pretty small place. The world's been definitely smaller the last couple of years, and the demands on the world for farming and food have expounded with the smaller size of the Earth. So we're looking at to produce more food, high quality food, desirable for the Earth's population. And the farming methods that we know of that have been working, that we're using, have been organic and conventional, and the biological farming, which we'll talk about today. So thanks again for coming. And so the first method of farming that I'd like to talk briefly about is conventional. Conventional kind of is set up from ever since it kind of got started, more or less to control or conquer nature. I mean, we use a lot of new high-tech innovations, the GMO seeds that insect-resistant, herbicide-resistant, the high inputs and dollar-wise, with chemicals and fertilizers to take care of these high-demanding new varieties of seeds is kind of, it can get to be a problem. The fertilizers keep going up, the seed keeps, the tech fees keep going up a little bit more, and it's a problem. But, however, it is potentially profitable. So the only issue that I can see right now is that the soil and food quality issues from conventional are having a bit of a problem. And that's why maybe a lot of people have tried organic. And organic works with nature. I mean, it has, limited inputs. You can only use certain natural fertilizers, pesticides, and seeds. It's all regulated. High management level. It takes a lot more management, if you will, to do organic farming. you have kind of limited markets. You always want to sell your organic produce or commodities or grain to your organic market, and sometimes those are not as easy to find as the county elevator. Potentially highly profitable institution, absolutely, and their soil and food quality would be one of their primary concerns. Now, biological works with all the natural laws. It's a systems approach using the best of both worlds, both methods. It's heavily versed in biology, physiology of plants, microbiology of the soil organisms. You have physics with energy considerations, and then we look at chemistry, the different chemistries of fertilizers, how they match, how they react. At first, when you first get started with the biological farming method, your inputs might be a little bit high at first, but it is potentially a profitable way to farm because the soil quality and the food quality are also our primary concern, our primary goal. So this photograph up on the top is a comparison between conventional and biological farming. 2 years ago in Osage, Iowa, we had plots. We had the same variety here shown, same hybrid. The one on the right with a massive root growth was the biological method of farming, and the conventional was just straight fertilizer, pesticides, whatever the local requirements were for that hybrid. So this was a pretty straight, good comparison. And the massive root growth is one of the things we see more often than anything else with the biological farming. Because the soil biology is the key. I had a professor once that said the soil microbes eat at the first table, plants eat at the second. I mean, that's just the way that Mother Nature designed it. All the nutrients must be transformed by the soil microbes into a usable form for plant uptake. This is like mineralization of nitrogen. They take the minerals, they convert them into their biology, their internal processes, making amino acids, making organic compounds, what they need to survive, and then when they die, those are transported into the plant in basically an organic form, an organic molecule. The organic guys don't like me to say that, but basically everybody is farming organically because it all has to go through the soil microbes. Now this video we're playing here is a close-up of a root as it's growing. The main root is on the left, and you can see the flow going up and down the root, and the little root hairs coming out from the side. This is a close-up of a root hair, and you can see that the plasma in the root hair is flowing, and there's little microbes, little dots on the outside of the root hair. And as we go into a closer look, what you're about to see is this area called the rhizosphere. Now the rhizosphere is this area in the sheath that's kind of surrounding the root as it grows, and you notice all those little dots in there are probably bacteria and soil microbes that are living in this sheath of the rhizosphere. And this is a symbiotic relationship between the microbes and the root. Now all roots leak. They leak sugars and starches coming up from photosynthesis, which comes down to the roots to feed it. And the microbes take these leaky sugars, starches, and plant metabolites, and they use them for food source. And then through the microbe transformation, they of course give back into the roots the nutrients and the minerals in a plant usable form. Also, there's like cells coming off those things on the right and left side of the root. Those are actually cells that are coming from the roots that the microbes then further, organic matter, if you will, start chewing on those and breaking those down into the nutrient elements that the plant can use. So this is the key. So in that previous picture, where you saw a massive root structure versus not so massive root structure from biological farming approach. The more roots you have, the more of these areas of the rhizosphere you have, so you're perpetuating more biology in the soil through this concept and reality of the rhizosphere. This is what I wanted to show you on that. So you must farm the soil microbes. They are the most valuable livestock on your farm. I mean, you need to increase their diversity, and you can do that through inoculation or rotation. You need to increase their food sources, and their preferred food sources, of course, the microbes really like organic matter. They're the key animals that break down the organic matter. Minerals, you can stimulate the microbes in the proper use of minerals in your soil, and of course they help break down and redistribute, recycle, if you will, the crop residue from your field. When this is all working well, they will in turn increase the water holding capacity of your soil, the gas exchange potential of your soil. You've got to remember that the roots need to breathe, and the microbes need to breathe. So there is gas. There's CO2 given off by the microbes, just like us. We exhaust CO2 when we breathe, and they need to pull in oxygen from the surface of the soil. So this has to have these open channels for gas flow, and those open channels, as they start to glue the soil particles together from polysaccharides that they kind of produce as their normal functions, they start gluing these tiny little parts of the soil together, and they start improving your texture. So those channels will absorb moisture from rainfall, so you're increasing all the benefits of your soil with a good, strong biology. The health of your soil will also be improved. You reduce the pathogenic outbreaks of root diseases, fusarium, those kind of organisms, because they just don't have room to grow in a very strong, highly diverse population of beneficial microbes. So all the crops stimulate or attract a specific group of soil microbes. Beans have a different soil biology than corn. So when you rotate, the rotation introduces or stimulates these groups of specific microbes, adding to the diversity of the soil biology or the soil biota. Many standard farming practices destroy or reduce the populations of these beneficials, and when that happens, they produce voids, where the voids are taken over by the pathogenic groups, and then pathogenic groups can proliferate and become a large player, if you will, in the soil biota. A lot of things that causes the voids are herbicides and fumigants, fungicides, overuse of anhydrous ammonia, excessive tillage. These are the kind of things that unfortunately, unknowingly, that we have a tendency to do, but it's not a beneficial aspect for the soil biology. A lot of herbicides are very, very strong chelators of minerals, and these chelators, even though you're not using that much, but you're getting some on the soil, they will chelate, hold on to the soil minerals, not allowing the microbes or the plants to obtain the nutrient benefits from the soil minerals. Another way that you can increase or get your soil biology going really strong, 100%, is you can inoculate. You can re-inoculate the beneficial soil microbes. That commercially, there's products that are essentially microbes in a jug. They will add diversity. They will increase the activity, and they will populate the voids previously created by inappropriate farming actions or farming management practices. Organic matter is a carbon source. We've all heard about carbon, how important it is to sequester carbon in your soil. A good carbon to nitrogen ratio is 20 to 1. At 20 to 1, 20 parts carbon to 1 part nitrogen, microbes easily break that down for food. And if it gets higher, say 40 to 1, it requires outside nitrogen for the microbes to get the energy to chew on this high level of carbon to nitrogen ratio. And these are like crop stubble, grass clippings, sawdust, things like that can get pretty high in the carbon nitrogen ratio. When that happens, in that case, you are actually slowing down the breakdown of the cellulose material in the residue, and this requires this requires nitrogen from some source outside in the soil. So what's happening to break down these higher rates of organic matter? You're stealing nitrogen from your soil. But when it is good, when it is happening, the microbes from the organic matter, they release sugars, enzymes, minerals, tied up fertilizer that just eventually break that material, that organic matter, into humus. And humus is about the smallest stable soil component derived from organic matter, and humus increases your water holding capacity. It's kind of like a sponge, if you will. The soil biology is attracted to the humus, so they proliferate and grow around the humus particles. It increases the soil texture by, holding water, by opening up those pores, and it's very, very beneficial to your overall fertility. So you need to keep that in mind. Those are the kind of the processes that organic matter breaking down goes through, and minerals is one of the most forgotten aspects of soil fertility. Minerals, just by themselves, will stimulate microbial activity. Calcium is one of the key elements, key minerals that seem to really have a big influence on microbial growth. We like to see about 2,000 pounds per acre at a minimum level of calcium, and that probably gets 75 to 80% of your base saturation. So calcium is really a key, but it also acts to help flocculate the soil. So it has a double-edged benefit here. The calcium does, whereas it's not only stimulating your microbial growth, but it's also physically and chemically improving your soil flocculation. Flocculation is just kind of a separation of tiny particles so that there's more air, and then they start forming little beds of soil. So it's a very, very good element to keep in mind. It's not just for regulating your pH. It goes well deeper than that. And the enzymes are very important. And enzymes are organic molecules produced by living cells that catalyze or speed up biological reactions. Now, every enzyme, which is an organic molecule, there is a mineral that is key in that chain, if you will. So with the enzyme in the chain, in the molecule, I mean the mineral in the chain of the enzyme, you have a complete functioning organic molecule enzyme, which then goes to the plant, goes to the cell, speeds up a reaction, makes a reaction, helps build the proteins. Whatever that enzyme's function is becomes complete when the mineral is in place. It is like a key for your automobile. I mean, if you don't have the key, your car's not going to start. If you don't have the mineral key for the enzyme will be complete. If deficient in minerals, these critical biological pathways will fail, or they will perform substandard. One of the examples that is very interesting is the schematic pathway. which is a biological pathway that is kind of like a callus, that when a fusarium or when a fungus tries to penetrate the cell, that it stimulates this pathway, and that pathway kind of cordons off or makes a callus around the hyphae that's trying to come into the cell, and keeps it from entering the cell. And that is a very, very good, very key defensive mechanism that a lot of plants have. And that pathway is one of the minerals that is key to the stimulation of that biological pathway, is manganese and phosphorus. And of course photosynthesis. I mean, the key minerals in that pathway are iron, magnesium, and phosphorus. So you have to keep in mind that even though your soil tests might show that you have some minerals, or your mineral levels in, say, iron or calcium are okay, are they bioavailable? And the bioavailability is why we use live microorganisms to inoculate the soil and make sure that those organisms are capable of creating those pathways with the key enzyme and the mineral. So crop residue is important, and it prevents erosion absolutely. Over the years, no-till and minimum till have greatly reduced the sediments flowing down the rivers and into the Mississippi, and taking our topsoil and giving it to Mexico. They also have a great important factor in holding and retaining moisture. I mean, with our residue on the top of the soil, of course you don't have the sun beating down on bare soil. You've got this kind of a buffer zone. You have a kind of a cover, and the moisture in the soil underneath this stays there, doesn't evaporate. So those are just two of the key things that we all pretty much are aware of. Crop residue must manage or return the nutrients to the soil for the microbial growth from the crop residue. I mean for instance, it's basically a ton of corn stalks contain 17 pounds of N and 4 pounds of P2O5, 34 pounds potassium, 35 pounds of calcium. And that's money in the bank. I mean, you have put that money in fertilizer in your crop, and you might as well get it back. Put it into the soil. And spraying lightly or incorporating a digesting fertilizer, a residue digesting fertilizer, will also increase the breakdown of your crop residue, and then you're starting to build humus, which we previously talked about is just black gold for your soil. You just can't really get enough of it. This is a kind of a close-up microscopic shot of after about 3 weeks of applying a residue digester on crop residue, lightly incorporating it, and you can see the fungal hyphae in there working on a root or a stalk, some type of cellulose material there. You have a fungal mat. I mean, these guys are they're like the first organisms that need to start working on that cellulose, those cell walls and stuff. So this happens pretty fast. So this is something that you need to consider and keep in mind, because without the microbial action, the crop residue breaks down very, very quickly. Energy is something that the last couple of years or so we've really been keying in on this, because all biological reactions either give off or consume energy. I mean, that's just physics. That's the way it works. These biological reactions, including ourselves, we're not stagnant. And one of the ways that we kind of keep a handle or keep a key on the energy is through the electroconductivity, or ERGs. And this is a measurement of the potential energy given off by 1 gram of soil per second. It is the potential capability of the soil to conduct electricity, and this is really important. I mean, the soil, think of the soil as a battery. like a car battery. I mean, it has to be able to conduct electricity or nothing will flow. You will get no ions flowing, you'll get no calcium, you'll get no magnesium, you'll get nothing flowing through the soil to get into the root, to get a breakdown, to have, say, like a calcium nitrate fertilizer break in half, break in the calcium, break into the nitrate molecule, and these have to flow into the root, into the rhizosphere, to be worked on by the microorganisms. So you need an electrical flow. If your readings are over 200 microohms per centimeter, there'll be no crop production. Uh, you want to get it down to ideally 150, 500 microohms per centimeter. This test, electrical conductivity, or ERGs, we call it ERGs, but a lot of people call it electrical conductivity, the same thing, is available for most agricultural labs. But what we have been playing around with last year or two or so is a method to determine the respiration of the soil. It's called the CO2 burst method. And this is a method that just measures the amount of carbon dioxide given off by a soil sample, which is directly indicates the total respiration of the soil biology. This is a simple test. The USDA uses it. The Soil Quality Institute uses it. 4,000 or 5,000 labs across the United States and Europe use this method. It's really simple. It takes 24 hours. And since the respiration can be an indicator of energy, because biology reactions indicate energy, there's energy there, this is an ideal test to determine the active energy level of the soil in real time. You get results in milligrams of CO2 carbon evolved, and these are directly proportional to the pounds of N mineralized available to you. So this is kind of a good double test here. You get the CO2 given off, which tells you the activity, and that number from this test is a pretty good indication, like 70% of the amount of free nitrogen that you'll have for your crop use provided to you by the soil microbes. 1,000 to 2,000 milligrams CO2 per kilogram of soil per week is the ideal range. And chemistry, fertilizers. Fertilizers are plant nutrients that stimulate at the proper life stages or cycle of crop. This is what we do. This is what we look at. This is what we formulate. All fertilizers are not the same. It's like, all cars have 4 tires and a steering wheel, but what's the difference between a Z28 Camaro and a Volkswagen? That's kind of the way that we're looking at formulating and blending our fertilizers. A lot of the teachings and directions that we've had in this area come from Kerry Reams, Dan Scow, Arden Anderson, Jackson, who wrote a phenomenal book on humates, Don Huber, Phil Callahan, Jeff Littrell, and Francis Chibosel, French agronomist that he wrote an excellent book in like the 80s, and it wasn't translated to English, and not until the 80s, but he wrote it earlier than that. And this is one of the things that he came up with was excess nitrates in the leaf attract insects. So these are kind of our guys that we have studied and talked with and helped us out. Plant growth regulators, or plant growth hormones, we like to use, and when properly used, can significantly affect the growth, the yield, and the quality of the crops. But all in all, the high energy fertilizers that release and absorb energy due to specific chemical and physical attractions of electromagnetic charges. I mean, this is just basic chemistry, physics, and we've taken this and applied it and made some excellent combinations of fertilizers. Now this chart here is real interesting and deserves a little bit of study, because what you're seeing is the difference between a male and female growth stage of a plant. On the left is the male side, and on the right is the female side. Now, one thing that they have to keep in mind is that the only thing that plants want to do is the reason why they're is to make seeds to carry on the next generations of their species. So when we're growing crops, we have a certain male stage and female stage. Now, to increase the male stage, which is early, which is vegetative stage, early growth, it's leafy vegetables are predominantly male, forage grasses, it's early season, and what can stimulate that in the physiology of the plant is things like calcium, potassium nitrate, chlorine. A little bit of chlorine can actually be very beneficial to stimulate vegetative or early growth, male-dominated growth. And this is kind of like a testosterone in human physiology. So, you've got a male testosterone plant that's growing. So how do you keep it there? How do you make it and full male, real strong? Auxins, gibberellins are some of the plant growth hormones that'll work in that case. And then as you get to where the plant starts to decide it wants to reproduce, to set seed, to make fruit, and fill the seed, you've got other formulations, other nutrients that will keep it and hold it in the female stage. So you're going to increase your fruit, you're going to make it hold better, you're going to increase your flowering, and by using sulfates, phosphorus, even a little bit of vinegar, ammonia nitrogen, NH4, not the nitrate nitrogen. Nitrate nitrogen produces vegetative growth, produces male growth. So you use the ammonial form of the nitrogen at this stage. It's very estrogen, very female, and cytokinins and ethylene production is what's going on. So looking at this chart, you could actually pop the plant when it's growing vegetatively in male stage. You could actually pop it with the proper foliar combinations and cause it to go into reproduction in female. I mean, and this is real kind of crucial for management stage, because if you have a short growing season like we do up here in Minnesota, you're not going to want that plant just to be lollygagging around and growing vegetatively for however long it wants. You just might have to actually force it into flowering and fruit set, because you can know how to do that now. So, what can you expect from a complete biological program? People have asked me that more than once, and I've been with the biological fertility game, if you will, for over 20 years. What can you expect? Well, you can expect increased plant vigor. You'll get darker color. The leaves will be greener. There'll be less yellowing. There'll be very, very few instances of visual signs of micronutrient deficiencies, yellowing, flashing, striping, those kind of situations. You will get more massive root structure, guaranteed, every time, without a doubt. Increased flowering. So increased flowering, you'll have more pods in soybeans, for instance. You're going to have more pods. You're going to have more seeds in those pods. So your yields will probably be significantly increased. You reduce disease and insect pressure because you're having a balanced nutrient approach. This Frenchman that I was mentioning earlier, Chabosso, his studies found that excessive nitrate nitrogen in the leaves that are waiting to be mineralized, or not mineralized, but metabolized into proteins, just waiting around, signal the insects. That reservoir of unused nitrate nitrogen in the leaves is a direct sign for the insects to come and have a smorgasbord, and that's what they do. So when you use biology, a biological program, you have a much more balanced and at proper times, nitrates, ammonia, nitrogen, you get this so you don't have the insect pressure, the soil, because it starts to open up and get texture developed, will absorb rainfall, and that rainfall won't run off. It won't be a hard surface, so you won't have excessive runoff. You won't have little ridges or rills in your field. You get heavier test weights from the final grain. Of course, you increased yield, but the best part about it, farming actually becomes fun. It's actually great to go out and walk your fields when everything is nice and green and there's no great problems. And you'll find this as you pursue or continue in a program of biological farming, that farming will actually start to become fun again. The products in the toolbox that we use, humates. Great, wonderful product. A biological product or fertilizer is one that either directly stimulates the biological growth or contains the live microorganisms in their byproducts. One of the most important ingredients in our program, and everybody is pretty much in agreement with this, is humic and fulvic acid. Now, the humates are a dry material. Well, what humates are basically they're dinosaur pastures. Uh, there's this great biomass thousands and thousands of years ago that were covered up by some geological process, an earthquake or something like that. And this biomass, this green dinosaur pastures, and even the dinosaurs too, probably, and all the bugs and insects, were just compressed through the geological process, and they were compressed and compacted and broken down ever so slowly, and they basically are this black, dark substance. And if that pressure continues, it will start to form soft coal and then harder coal. Another name of this is Leonardites. So that's kind of where humates came from, high in carbon, and the most usable or the immediate usable aspects coming from humates are fulvic and humic acid. They can be directly used compounds solubilized by from the humate substance. They're huge organic molecules, and they've been known to have numerous plant-stimulating effects, contain a vast array of bioavailable minerals, an excellent source of biological carbon, have been studied by various people and researchers since the 60s, and the work and the investigation is still turning up more amazing plant growth and mineral holding capabilities of the humates. Um, all biological products that work contain humic or fulvic acid or both. And William Jackson, who I mentioned earlier, wrote a phenomenal book on humic fulvic acid, and it's called Humic, Fulvic, and Microbial Balance Organic Soil Conditioning. If you need a really good book stop, or you are an avid reader, you could get a hold of this book. It's about 6 inches thick, and it'll kind of open up your mind to the humates and the fulvic and humic acid. So, our soil applied products. Our object is to inoculate, stimulate, and feed the vast array of beneficial soil microbes. They should contain, of course, humic, fulvic acids, carbohydrates, minerals. One of our products is MicroPack. It contains over 125 indigenous beneficial species of soil microorganisms, seaweed kelp, bioorganic catalysts, and it works very well with our high energy fertilizers. Some of the other examples of soil applied products are contain calcium, sulfur, ammonia, humic and fulvic acid. Calzol is one. We have a strip-till mix we call. residue digester is another one, and they all have the humic fulvic and or combination, so they all work very well. The most important aspect of biological farming and farming in general, but is to always have adequate nutrition for your crop in your crop. Uh, and this, takes a lot of management. It can be viewed as spoon feeding. I'll grant you that. But can you eat a whole year's worth of food at one sitting? Well, neither can a crop. Neither can a plant. I mean, you can't dump the entire regime of nutrients that this plant needs for make 200 bushel corn, dump it all there and expect it to be fine. So we are firm believers in foliar nutrition. Foliar nutrition is something immediately absorbed through the plant leaf if you have the right formulation. For those of you that think that spraying something on the leaf doesn't work, a waste of time, uh, how do herbicides work? It's the same thing. Foliar is used to apply critical nutrition. It's used to stimulate the crop from one stage to another, as we discussed earlier. You can salvage a stress crop from a drought or hail, insect disease, and retain and finish the grain, improve your test weight. These are some of the examples of some of the products that we have. Defender G4 is a trace mineral with a great deal of essential minerals that the plant needs, particularly at early growth. The stimulate is one that we use as mostly plant growth regulators. It uses the plant growth regulators to stimulate the plant to branch, for one thing. It stimulates branching in soybeans a great deal. And salvage, one of the salvage products that we can use or have used is copperite, which is nothing but a 7.5% copper with phosphite, and that has a tendency to boost the copper in the plant, which is kind of aid in stimulating the key biopathways that reduce disease and insect pressure. An example of the foliar program on corn, we start early with the foliar stimulate and the Defender G4, which is a mineral package. You need to get these minerals in there early in the plant to make sure that those biopathways and those enzymes have those key mineral keys to make that biology, make that biopathway work at 100% efficiency. We also come a little bit later. You notice that there's a sidedress involved here. So when you foliar, you need a good source of ground fertilizer, pre-tassel, and a late season foliar on corn with the seed set, and our late season bulker rondo. Kind of the same situation in soybeans. We early foliar it with the Defender G4 minerals. Pre-flower, we hit it with the seed set to start setting those flowers, to start holding those pods, and you can do this a couple of times throughout the year, and then we end up with the late season bulker. This is just an example of some of the data that we've had over a couple of years, and really what I want to show you more than anything else is that as individually, say like the micropack in the first column or the first row, by itself is only maybe 3 or 4 bushel increase. But when you start combining the micropack, the next column, next row, with the high energy fertilizer formulation of the 8-19-3, you have tremendous benefits, tremendous increase in yields, which is the same further down with the Biomax, the Defender G4, and the other high energy fertilizer 1797. So this is kind of an example of some of the things we've been isolating, some of these key variables, and we get some very, very good results. The conditions that will adversely affect biologicals, well, human error. I mean, we can, not put it on properly, not at the right time, wrong time of day. You really have to want this to work is one of the big factors in human error. If you decide it's not going to work, you're just out there wasting your time. Mixing it with sides, fungicides, herbicides, very cautious on that because the sides kill microorganisms. So you have to be careful if you tank mix. Waterlogged soils, it does not work well in waterlogged soils. You need drainage, you need that oxygen, you need that gas flow. And poor quality water will affect all chemicals, will affect everything. and you really should seriously consider using an RO source of water, because your chemistry is going to work so much better. Um, the real world of FHR Farms. FHR Farms is the company that is hosting this, and they farm from Spring Valley to Chatfield, Blooming Prairie to New Richards, southeast Minnesota. We got 1,400 acres of corn, corn on beans. We got about 300. Do a little bit of grass, alfalfa hay. Total about 2,300 acres of farming, custom farming with the biological program, and 130 cow-calf herd who is fed directly off of our biological farming, so they're not eating GMO feed. Nutrient management. High growers for growers is our kind of byline. We're in fertilizer manufacturing, distribution. We have a RAL research aspect that does plots across 9 states, comparing non-GMO varieties with the U-Trough method. We have CSI Laboratories, which is just starting to come on, designed to test for glyphosate and the breakdown products AMPA. High Yield Transportation is a trucking, semi-trucking outfit that we use to transport all our nutrients around the globe, around the country. In 2014, this is very, very good. We formed a seed company with, we will be producing 100% non-GMO seed that is going to bring genetic purity and the highest nutrient density to the consumer. So we're real excited about that. And coming soon is CSI Laboratories. This will be our laboratory that's under construction now, and it will test for glyphosate, a breakdown product, AMPA, and 2,4-D. And if you're interested and you need these services, just contact the FHR website. It's fhr1.com for the updating status of the lab. So kind of in a little bit of summary, we have a systems approach that we have been using for years, and getting all these things squared away, understood, and applied. So we're starting with science. We apply that knowledge into the manufacturing of fertilizers, and then we apply that product with precision nutrient placement. And we have some high-tech platforms that we use. One of the things that, of course, that we use and we found very effective is strip-till, which is precision placement of your seed, your fertilizer, and the location comes back to the same place every time, so you need to help you locate that. And down here, the strip-till is the... Now we've taken that step forward, and we've come to precision with a U-Trough twist. And the only guy that I know that can very well explain the U-Trough is Jeff Littrell, one of the designers and brains of the U-Trough system. So Jeff, are you there?
Yes, yes I am. So what we're going to do, thank you John, thank you Daryl, and thank you to all the attendees. We're going to show you now the planter that Keith Schlapko and myself actually designed. Keith actually built the first one. This video is somewhat, you can go online and be able to pick this up. So what I'm going to do is read through it very briefly to help you folks or explain it. When we designed the planter, the idea came from the South African group that was actually building a planter that put liquid on one side, and I should say dry on one side, dry on the other, with liquid attachments, and they're keeping the calcium and the sulfur apart because they could not get it to mix and blend. When Keith and I looked at it, we figured out one of the products we've already been using. As you can see there, the coulter, we're putting down the combination of ammonium nitrate, ammonium sulfate, phosphoric acid, potassium sulfate, calcium sulfur, our micronutrient package, all in a homogeneous form that's built in the Midwest. And then we actually take that and we run that through on the one side of the planter, and then as you'll see coming up here shortly, on the opposite side of the planter, in another John Deere coulter. Both these are John Deere coulter with gauge wheels on them. We combine your 32 or 28% with our product called Calzol, which is a combination of thiazole and calcium nitrate with humates, and basically our patented water system that we use to build all our fertilizer with. So we take that, and what you're doing is when you're welding on the farm, you have to have a certain level you're from that piece of metal, so you get a good arc. That same thing happens within the soil system. We want to have a good arc so we can actually maintain that. And then below the seed, we have that 819-3, the amazing starter with the micropack thrown in there, and we actually create a U-Trough. So that way we're building a seed system that develops all the way around that plant. So that way you can have an attainable operation like we have. So in the long run, what we're trying to do is actually build a system that will make my rented farms as well as our own ground, our custom farm, so we can maintain that level long term. John, back to you to conclude things up. I think we're getting
thank you
close.
very much. An explanation. This is our U-Trough system working in the field. The Montag dry fertilizer tank on the back to apply the dry fertilizers, and this is kind of what it looks like strip till situation.
Basically what we're doing there, I'll interject again quick, John, as we're going across the field of SideQuest tanks, we're putting down our 415-8 liquid, and then the dry is a combination of the soft rock, 819-3, and then basically mix and stirs that into that zone, into a V, so that way we come up with without doing a lot of disturbance, and we put our digester up front, spraying down that residue and then sizing it. So our goal is to spray the residue digester and actually size the stock, apply the nutrients, as you can see here in the dry system, and then still come back in and close it up and do the best we can to make a good zone for the coming spring. As you can see there, 240-pound soft rock, 20 of MAP, 20 SOP, 20 of KCL, and then basically 415-8 liquid injected in that zone with humates. John?
This is basically the composition mapping, which we probably won't key on very much, but you need to know what you need, know where you put in the tractor cab, which is real-time adjust. You can adjust things on the fly. Some of the emails that we've got from downloading this information to our iPad, and you can see that that strip till in the bottom half of field B is far and above the 175 bushel conventional tillage field. This is one of the access from the information. This yield map, and this green is not good. The red is up to yield, so you have you can see field where you need to do some work and where you need to improve on, because it's versus your field where you're losing yield. This is an interesting field here. There's corn on beans and a corn on corn split through on that white line. You see that big on the left field. Well, we've got some serious problems, issues there. Red is bad. So, you know, even though we had an average yield of 201 bushels, what is going on in a big red spot? Well, we took some aerial photography for it, and it's basically a drainage problem. You see in that same field, aerial photography, you see that it's light, and if you really blow that up, you can see that the top that were laid there are up and not effective. So one key issue to using an airplane and using these mapping programs, you can get exactly what's going on. This is a precision dual-purpose applicator. It's a sprayer and a ground fertilizer rig. This is developed by Keith Schlopko down in southeast Iowa. It has spray nozzles on the top and has kind of a dribbler end applicator on the bottom, has an angle with hoses dripping out. And if you notice that he can put down nitrogen in this picture at the lower right there, right at the stalk of the corn, right at the base of the corn. He can dribble nitrogen and foliar apply nutrients at the same time, and he puts it that close to the stalk, because even in a dew, you will get the moisture from the dew coming down the stalk and giving you a 2 or 3 inch diameter moisture zone around your plant even during a drought. So, your nitrogen that you're applying is being highly solubilized. This is the walker in action with the spray nozzles. And I would just like to conclude this brief introduction by saying that biological farming is the best of both worlds, perhaps the pinnacle of farming knowledge. It's adaptable. It allows your farm to adjust to the seasonal changes. It will buffer and increase your soil's productivity. It will also allow a world that is reachable at your fingertips. All it takes is desire and a keen eye. See what you look at, observe, and pay attention. The changes at first are subtle, but they are just the beginning. And your family. It's a system that holds the promise for soil reclamation, higher quality food, increased yields, and over time, it has been around since the beginning of agriculture, but we just need it now more than ever. And I'd like to throw up this reading list. Our world have helped us along and got us keyed in. You can further follow along. These books, they are phenomenal. They're very interesting. If you want some real-time information, though, you can go to the website at www.f1.com. You can download or read the inputs, the fertility programs, get a lot more information. See some videos of some key speakers that we've had at our seminars. And by the way, everyone's invited to our seminars when they come rolling around. And kind of see if we have any questions.
Sure, John. Thank you very much for that. And feel free to leave that screen up there with some of the reading material while we go through the questions.
Absolutely.
We've got just a few minutes here. We'll try and get through as many questions as we can. And if there's some that we're not able to answer, we're going to kick them back to John and Jeff, and they can provide us some written answers that we can put up on our website when we place the archived webinar on the website. A couple questions about the carbon dioxide and also CO2 burst. One question is, what time of year should you test the CO2?
At any time of the year. You can test it early in the spring. You can test it mid-season, but this is right. So what you'll be doing is you'll be seeing the activity level. I mean, it would of course be ideal to have that activity level good throughout the year. But if your funds are a little bit limited, take one time of the year to test it. It would probably be just after the soils warm up, maybe right at planting time, or just after you see the ground.
Come on.
And that would be a good time to see how strong your biology is going to be and how much nitrogen that you will be. Is there anything else?
Jeff, I think you wanted to add something to that as far as when you do it. Okay, I'm gonna go to the next question here. With CO2 bursts, in regards to measuring the total, how can you know qualitatively that the bios are beneficial? So as you're doing these measurements, how do you know what you got there is beneficial?
Test has been is from Woods End Laboratory in Maine, and it has been highly calibrated, and through the development of their test, it's color indicator that changes inside of a jar. You put the soil in a jar, about like a quart jar, and you add a little bit of water, direct relation to the amount of soil, 40 grams of soil and 20 mils of water, and this brings the soil to fill capacity. And then you incubate it with this color indicator in there for 24 hours. The history of the color change, and the amount of CO2 given off, captured by that color indicator, highly correlated and researched. Peer reviews have been written on this methodology, so it's been all calibrated and correlated by the company that produces these meters and the soil test. So for the end user, it's real simple. And you take your color indicator, you put it in, and it gives you a number, and then this number, it's the CO2 evolution sample, and then your chart that they have done extensive research on to calibrate the amount of CO2 versus the amount of N mineralized. So that's where that comes from. Do a whole lot of thinking about. You just have to follow the simple procedure and use their meter to read it. And it's I mean, there's a lot of soil labs out there that are using this and charging you to $25, $35 for this. It supersedes the old test of basically killing, sterilizing this soil, and then going back and figuring out the way of the in there. It's kind of messy, and so this test is very straightforward, very correlated and calibrated. So I have a 100% confidence that this test is giving you accurate numbers.
Okay. This is an interesting question that once a soil has recovered a good biology, and through working with a system like you've got with the U-Trough, and if you get the biology in stable nutrient pool where you want it to be, how would that could you go without a U-Trough system, or maybe it's more how would you modify it? I mean, how, Jeff, maybe you can talk about through the years as you've worked with this system, maybe how you've modified what you might put down with a planter as you feel like you might be getting your soil biology working for you.
Here's the thing that I guess that I've learned over the years from my mentor that had passed away recently, Dr. Dan Scow, and all the other folks, is that as long as we're using sides, we are going to keep having to figure out how to work with biology. There's a ton of people that say, oh, it's there, we just got to activate it. I don't care what biology, what type of herbicide we're talking, the biology is going to be fragmented or reduced. So by doing this system, we have found if we're going to keep doing sides, which I do, we're going to keep putting biology in. But back to the question on the U-Trough, we've got folks that are using a single coulter system with the in-furrow, that strip-till, single coulter. I'm not saying that U-Trough is the only way of doing this. For our operation and our farming setup, where we're renting 60% of our ground plus, we're custom farming about 48% of our ground. This makes the most sense for someone in that realm that is custom or is actually renting a large chunk of their acres, because we can only control that year or 2 years, however long we have that contract. So this made more sense to us. You own all your ground. God bless that. I think it's a great thing. I think then you're going to have to come. You can come in. You could bulk spread, and you could everything you do is going to maintain higher levels. That's what Reams taught us. Scow's mentor, the creator of the biological theory of ionization, is that you spread it all across the farm, and you create a grainer, a magnetic field, all these wonderful things that go along with that. But in the real world in 2013, that's impossible. We don't have enough dollars left. When Reams taught this to Scow and so on and so forth, they were dealing with under $100 a ton potash, $125 a ton MAP, and $70 a ton 28%. Everybody knows where we're at now today. It's a whole lot bigger environment. So I would say you could do however you want to modify this. There's no set way. We have found this to be a system that works on that farm. It's a little more spendy, but at the end of the day, I think you're going to create a better root zone, a better, healthier program. To quote Bob Strike, good friend of Keith and I's, the more straws in the soup, the more you drink. So if you can create that through that biological U-Trough system, you're going to create a much healthier long-term approach to your farming operation, Daryl.
All right. Thank you very much for that, Jeff. I think we're going to close the webinar at this point, and we do have some other questions. We're going to forward those to both John and Jeff, and so when they have some time to sit down and answer those, and we can share those up on our No-Till Farmer website along with the archived webinar. As I wrap things up today, just a quick note that, again, we will be making an archived version of today's webinar available on the website at no-tillfarmer.com. Once we have that replay ready, we will send out an email to all of you who attended today with a link to the presentation. So you have that opportunity to view it and listen to parts of it again if you'd like to. And again, we will get further questions and answers up there as well. So I really want to thank you all for joining us again today in this webinar. I want to thank John and Jeff for presenting as well, and FHR for sponsoring this one. Also, thanks to my No-Till Farmer staff here. I got Cole working here, kind of the producer, so to speak. And so I wish you guys all a great afternoon, and we will hopefully be able to talk to you again real soon with another future webinar. Have a great day.
Thank you.
Thank you.